Darwin's finches are a group of 14 species of small birds, 13 of which occur on the Galapagos Islands. This group of birds provide one of the most striking and best studied examples of evolution on islands. The Galapagos Islands are a particularly striking natural laboratory of evolution. The islands are all relatively young in geological terms, and they have never been connected with the adjacent mainland of South America or with any other source area. All of the organisms that occur in these islands have reached them by crossing the sea as a result of chance dispersal in the air, water, or by air, wind, or by transport via other organisms. Since the Galapagos are a group of islands, small flocks of birds could occasionally reach new islands where no other land birds were present. On the new islands, the immigrants would become adjusted to the local conditions through the process of natural selection. In doing so, they would have become progressively more different from the original population. The descendants of the original finches that reached the Galapagos Island now occupy many different kinds of habitats in the island. These habitats encompass a variety of niches comparable to those occupied by several distinct groups of birds on the mainland. Among the 13 species of Darwin finches that inhabit the Galapagos, there are three main groups. The three groups are ground finches, the 6 species of ground finches. Most of the ground finches feed on seeds of different sizes. The size of their bills is relative to the size of the seeds on which the birds feed. Then tree finches. There are 6 species of tree finches belonging to several diverse groups. These species also differ from another primarily in bill size and shape, which reflect adaptations to their food. There are 6 species of tree finches belonging to the several diverse groups. The species also differ from one another primarily in bill size and shape, which reflect adaptation to their food. 4 species have bills that are suitable for feeding on insects, and one with larger bills feed on larger insects, and those 4 with smaller bills feed on smaller insects. One of the tree finches have parrot-like beak. It feeds on buds and fruits in trees. The 6th species of tree finch, the woodpecker, finches a chisel-like beak. This unusual bird carries around a twig of or a cactus spine, which is used to probe for insects in deep recesses. It is an extraordinary example of bird that uses a tool. Then Warbler's finch, this unusual bird differs from the ground finches and the tree finches in the habitats and morphological adaptations. The warbler finch plays the same ecological role in Galapagos woods that warblers play on a mainland, searching continuously over the leaves and branches for insects. It has a slender warbler-like beak. So this picture shows 10 species of Darwin's finches showing differences in bills and feeding habits. The bills of several of the species resemble those of different distinct families of birds on the mainland, a condition that presumably arose when the finches evolved new species in habitats where other kinds of birds normally occur on the mainland and which are not available to finches here. Vegetarian tree finch, fruit eaters, large insectivorous tree finch, small insectivorous tree finch, then woodpecker finch, then warbler finch, cactus ground finch, sharp-beaked ground finch, small ground finch, medium ground finch, large ground finch. Tree finches, warbler finch, ground finches, then grassling bills, insect eaters, fruit eaters, robbing bills, cactus eaters, seed eaters, crushing bills, ground finches, tree finches, and warbler finches. To Darwin, it appeared that each was slightly modified from an original colonist, probably the finch on the mainland of South America, some 600 million to the east. It is probable that adaptive radiation led to the formation of so many species because other birds were few or absent, leaving empty niches to fill. And because the numerous islands of the Galapagos provides ample opportunities for geographic isolation. Types of natural selection. If an individual has same form of trait that gives him a better chance of survival in a given environment, eventually his offsprings inherit the trait. This is known as differential survival. Differential survival would refer to the survival of individuals in an environment to which they are well developed or well adapted. The adaptations are genetically controlled and heritable, So differential survival leads to differential reproduction of alleles responsible for the survival of the next generation. And natural selection means differential reproduction among members of the same gene pool. Types of natural selection: directional selection, stabilizing selection, disruptive selection. Environmental changes don't necessarily induce genetic changes. The genetic variants composing a species may be sufficiently homeostatic so that adaptiveness remains adequate in the altered environments. The Darwinian fitness of variant phenotypes and genotypes may be shifted, some in positive and others in negative directions. A directional natural selection then comes into operation. It transforms the gene pool of a species or population toward the highest level of adaptiveness that can be reached in the new environment. Under stabilizing selection, extreme varieties from both ends of the frequency distribution are eliminated. The frequency distribution looks exactly as it did in the generation before. A real-life example is that of the birth weight of a human baby. Features of stabilizing selection: operate in constant or unchanging environment, keeps a population genetically constant, favors the average or normal phenotypes, introduce homozygosity in the population, then check accumulation of mutation. Then under directional selection, individuals at one end of the frequency distribution do especially well, and so the frequency distribution of the trait in the subsequent generation is shifted from where it was in the parental generation. Industrial melanism is such an example. So features of directional selection: due to the changes in the environment in particular direction, favors the phenotype which is non-average or extreme, then alters the mean value of the trait in the population in one direction, favors accumulation of those mutations that increase fitness in the changing environment, then eliminate normal or average individuals. Under diversifying or disruptive function selection, both extremes are favored at the expense of intermediate variants. This is uncommon, but of theoretical interest. Both extremes are favored at the expense of intermediate varieties. This is diversifying or a disruptive selection. Earlier, we learned about directional selection, then disruptive selection. It is uncommon but theoretical interest. It is of theoretical interest because it suggests a mechanism for species formation without geographic isolation. The features of disruptive selection: previously homologous population break up into several different adaptive forms. Extreme value have highest fitness and intermediate or mean value are relatively disadvantageous. It occurs when a population previously adapted to a non-homologous environment is subjected to divergent selection pressure in different parts of its distributional area. Here we could see a picture which depicts all this. Directional, stabilizing and disruptive selection. First, this one is the original parent phenotype, original distribution. Frequency, y-axis, phenotype, x-axis. For example, height. The mean value is also represented. Frequency on the y-axis, phenotype, height and mean. First is about directional selection. Directional selection before selection, like this. Directional selection. And extreme is favored. Then stabilizing selection. Both extremes are favored. Disruptive selection, only the middle one is. That means excluding both the extremities. Then after selection, it will be like this. Okay. Directional, stabilizing and disruptive selection. Shaded areas show the groups being selected against. At the top is the original distribution of individuals. The final distribution after selection appears in the bottom row. Pattern of evolution. Convergent evolution and divergent evolution. We often find unrelated animals converging on the same form of structure. Pattern of evolution, convergent evolution and divergent evolution. We often find unrelated animals converging on the same form of structure because that form is very adaptive in their common environment. The special case of evolution is called convergent evolution. Example of convergent evolution is the streamlined shape of sharks and dolphins. One is a fish and other is a mammal, and they are related to one another in only the most distant sense. But if your life depends on swift movements through the water, then a streamlined shape is pretty much essential. Another example is wings in birds, bats, and flies. So convergent evolution produces analogous structures. Then divergent evolution is the process of two or more related species becoming more and more dissimilar. The red fox and the kit fox provide an example of species that have undergone divergent evolution. Divergent evolution produces homologous structures. The same bones can be used in many ways, leading to several divergent evolutionary paths. Frogs, bats, birds, men, and so on. Then homoplasy. Homoplasy refers to the similarity among unrelated organisms. Similarity among unrelated organisms. Wings of birds, bats, and insects are homoplastic features because the common ancestor of these three groups lacked wings. Spines, modified leaves, and tons, modified stems are examples of homoplasy among plants. Spines and tons look similar and both provide protection to the plant, but the plants to which they belong are distantly related. Homoplastic similarity can be anatomical, physiological, or behavioral. Then coevolution. The term coevolution is used to describe cases where two or more species reciprocally affect each other's evolution. For example, an evolutionary change in the morphology of a plant might affect the morphology of an herbivore that eats the plant, which in turn might affect the evolution of the plant, which might affect the evolution of the herbivore, and so on. So, coevolution is likely to happen when different species have a close ecological interactions with each other. These ecological relationships include some aspects of predator prey and parasite host relations, competitive species, mutualistic species. Plants and insects represent a classic case of coevolution, but that is often, but not always, mutualistic. Many plants and their pollinators are so reliant on one another, and their relationships are so exclusive that biologists have good reason to think that a match between the two is the result of a co-evolutionary process. But we can see exclusive matches between plants and insects even when pollination is not involved. Some Central American acacia species have holotones and pores at the bases of their leaves that secrete nectar. These holotones are the exclusive nest site of some species of ant that drink the nectar. But the ants are not just taking advantage of the plant, they are also defending this acacia plant against herbivores. The system is probably the product of coevolution. The plants would not have evolved holotones of nectar pores unless their evolution had been affected by the ants, and the ants would not have evolved herbivore defense mechanisms or behaviors unless their evolution had been affected by the plants. And parallel evolution. Parallel evolution is the development of similarities in separate but related evolutionary lineages through the operation of similar selective forces on or similar selective factors on both lines. Parallel evolution is similar to convergent evolution in that similar adaptations arise in different species. The main difference is that parallel evolution, the two species have a relatively recent common ancestor. Then adaptive radiation, natural selection, can ultimately lead to the formation of new species. Sometimes many species evolve from a single ancestor species. Similarities in skeletal and muscular structure of Hawaiian honey creepers lead scientists to conclude that the 23 species of honey creepers evolved from one ancestor species. Such an evolutionary pattern in which many related species evolve from a single ancestor species is called adaptive radiation. Adaptive radiation most commonly occurs when a species of organisms successfully invades an isolated region where few competing species exist. In Darwinian finches, there are 14 species of finches in the Galapagos Islands, each filling a different niche on different islands. All of them evolved from one ancestral species which colonizes the islands only a few million years ago. This process, whereby species evolve rapidly to exploit empty ecospace, is also present in an example of adaptive radiation. Once again, adaptive radiation I'm going to read. Natural selection can ultimately lead to the formation of a new species. Sometimes many species evolve from a single ancestral species. Similarities in skeletal and muscular structure of Hawaiian honey creepers lead scientists to conclude that 23 species of honey creepers evolved from one ancestral species. Such an evolutionary pattern in which many related species evolved from a single ancestral species is called adaptive radiation. Adaptive radiation most commonly occurs when a species of organism successfully invade an isolated region where few competing species exist. So, if new habitats are available, new species will evolve. There are 14 species of finches on the Galapagos Islands, each filling a different niche on different islands. All of them evolved from one ancestral species which colonized islands only a few million years ago. This process, whereby species evolved rapidly to exploit empty landscape, spaces also present an example of adaptive relation. Then species and speciation. What is a species? Biological species concept. The biological species concept put forward the Mayer defines a species as members of populations that actually or potentially inbreed in nature, not according to similarity or appearance. Although appearance is helpful in identifying species, it does not define species. This concept emphasizes that a species is an evolutionary unit. Members share genes with other members of their species and not with members of other species. Members share genes with other members of their species and not with members of other species. Okay. So phenetic species concept has species as a set of organisms that are phenotypically similar and that look different from other sets of organisms. According to this concept, phylogenetic similarity is all that matters in recognizing separate species. For example, if the two frogs look same, even though they are prevented from mating with each other, they would be considered the same species according to the phenetic species concept. Then evolutionary, then phylogenetic species concept, evolutionary or phylogenetic species concept. George Gillard Simpson defines an evolutionary phylogenetic species as a lineage, an ancestral descendant sequence of population evolving separately from others and with its own evolutionary role and tendencies. Evolutionary lineage is often conveniently represent a tree-like diagram called a cladogram. Construction of evolutionary tree is termed cladistics. Understanding of phylogeny is a lot like reading a family tree. So the root of the tree represent the ancestral lineage and the tips of the branches represent the descendants of that ancestor. As you move from the root to the tips, you are moving towards in time. When a lineage splits, that represents speciation. It is represented as branching on a phylogeny. When a speciation event occurs, a single ancestral lineage gives rise to two or more daughter lineages. Descendants, ancestor, descent, past. In the above figure, bifurcations represent products of the speciation process, where one species becomes subdivided into two or more groups that follow their own evolutionary ways to become new species, coexisting at the same time. So, certainly different branches of a cladogram reflect the idea that these populations are considered to have evolved separately with their own evolutionary rules and tendencies. A group of species derived from a single common ancestor population is said to have a monophyletic origin. A group of species classified together, some members of which are descended from different ancestor populations, is said to have a polyphyletic origin. A species consists of a single uniform population devoid of differentiated and semi-isolated local populations, races. Subspecies is monotypic. A species which consists of a single uniform population devoid of differentiated and semi-isolated local populations is monotypic. A species of polytypic if it is subdivided into two or more race or subspecies. A population or species that show no genetic or phylogenetic variation for a particular gene or character is termed monomorphic. The presence of two or more genetic or phylotypic variation in a population for a particular gene is described polymorphism. Then race, a population or group of populations of a species that share a geographically and or ecologically identifiable origin and have unique gene frequencies and phenotypic characters that distinguish them from other races. Because of the large amount of genetic and phenotypic variability in most species, the number of racial distributions that can be made is often arbitrary. Then sibling species so similar to each other morphologically that they are difficult to distinguish, but that are nevertheless reproductively isolated, sometimes called cryptic species. Similar to each other morphologically, difficult to distinguish, but are nevertheless reproductively isolated. Then subspecies, a taxonomic subdivision of a species often distinguished by special phenotypic characters and by its origin or localization in a given geographical region. Like other species subdivisions, subspecies can still interbreed successfully with the remainder of the species. However, in some cases, interbreed capabilities are unknown and subspecies designations, Homo sapiens, Neanderthalensis, and Homo sapiens are based entirely on phenotype. And speciation is a lineage splitting event that occurs two or more separate species. It occurs when gene flow is reduced sufficiently between sister populations to allow each to become irrevocably committed to different evolutionary lineages. Imagine that you are looking at a tip of the tree of life that constitute a species of fruit fly. Move down the phylogeny to where your fruit fly twig is connected to the rest of the tree. That branching point, every other branching point on the tree, is a speciation event. At that point, genetic changes result in two separate fruit fly lineages where previously there had just been one lineage. But why and now did it happen? Diagram representing speciation events in genus Drosophila. Speciation events, various kinds of Drosophila. So, anagenesis versus cladogenesis. Most evolution consists of two processes, anagenesis, also called phyletic speciation, and cladogenesis. Phyletic evolution is the progressive evolution of species involved in a gradual change in gene frequency in the entire population. Phyletic evolution or anagenesis. Anagenesis is a progressive evolution of species involving a gradual change in gene frequency in the entire population. When enough mutations reach fixation in a population, so slightly differentiate from an ancestral population, a new species name may be assigned. A key point is that entire population is different from the ancestral population, so that the ancestral population can be considered extinct. When a species is transformed over time by the acquisition of phenotypic and genetic modifications, there is no increase in the number of species. And speciation results in the splitting of lineage into two or more species is described as cladogenesis. Species results in the splitting of lineage into two or more species is described as cladogenesis that subsequently diverge in their traits through anagenesis. So species 1, 2, 3 like that. Forms of speciation in cladogenesis, which is represented with the alphabet A. A species changes over time until it is so different from its progenitor that it is classified as a new species. In cladogenesis, speciation takes place as a branching process when one species becomes two or more. Examples of speciation. Jane Root and her colleagues studied the evolution of digestive physiology in Drosophila, and they collected flies from a wild population and established which one? Drosophila pseudo obscura. So they collected flies from wild population and established several separate laboratory populations. Some of the laboratory populations were raised on starch-based medium and others on maltose-based medium. Both food sources were stressful for the flies. It was only after several months of evolution by natural selection that the population became adapted to their artificial diets and began to thrive. Rhodes' team wanted to know whether the starch adapted populations and the maltose adapted populations, which diverged under strong selection and in the absence of gene flow, had become different species. Roughly a year after the populations were established, a series of mating trials were performed. For each trial, 48 flies were placed in a bottle, 12 males and 12 females from a starch adapted population and 12 males and 12 females from a maltose adapted population. The researchers then noted which flies mated. So, Dodd's group predicted that if the populations adapted to different media have speciated, if populations adapted to different media have speciated, then the flies would prefer to mate with members of their own population. If the population had not speciated, then the flies would mate at random. So roughly 600 of 900 matings observed were between males and females from their own population, like same population. In other words, the differentially adapted fly population showed partial pre-mating isolation. Dodd and her colleagues concluded that the population had begun to speciate but had not yet completed the process. Incident speciation in laboratory populations of Drosophila pseudo obscura. Number of matings involved each kind of male-female pair. You could see this. Then mechanism of cladogenesis. Reproductive isolation. There are many barriers to reproduction that involve during the course of speciation that limit gene flow between sister species. Each species may have its own courtship displays or breeding season so that members of the two species do not have the opportunity to interbreed, and the two species may be unable to interbreed successfully because of failure of egg to become fertilized or to develop. This suggests a simple and useful dichotomy between pre-mating or pre-zygote formation, reproductive isolator mechanisms, and post-mating or post-zygotic isolating mechanisms. Pre-zygotic isolating mechanisms prevent the formation of hybrid zygotes. Ecological isolation. Species occupy different habitats. Temporal isolation. Species breed at different times. Then behavioral isolation. Species engage in a distinct courtship and mating rituals. And mechanical isolation. Mechanical isolation in the sense interbreeding is prevented by structural or molecular blockage of the formation of the zygote. Mechanisms involve inability of sperms to bind to the egg in animals. Other female reproductive organism of a plant is preventing the wrong pollinator from landing. These are some pre-zygotic isolation mechanisms for cladogenesis, which are the ecological isolation, temporal isolation, behavioral isolation, and mechanical isolation. Then post-zygotic isolation mechanism. Post-zygotic mechanisms are those in which hybrid zygotes fail, develop abnormally, or cannot self-reproduce and establish viable populations in nature. Hybrid invariability. Hybrid invariability. Post-hybrid invariability in the sense development of zygote proceeds normally, but the hybrid does not survive. Hybrid inviability, not viable. Then hybrid sterility. The hybrid is healthy but sterile. The mule, the hybrid offspring of a donkey and a horse, mare, is sterile. It is unable to produce viable offspring. Donkey and mare. Then hybrid is a healthy and fertile but less fit, or infertility appears in later generations, as witnessed in laboratory process of fruit flies, where the offspring of second generation hybrids are weak and usually cannot produce viable offsprings. Then allopatric, parapatric, and sympatric speciation. Reproductive isolating mechanisms are barriers to gene flow, the spread of genes between populations. These isolating mechanisms can evolve in three different ways, each of which defines a different mechanism of speciation. In 1963, Ernest Mayr contended that speciation takes place through allopatry or geographic isolation. Allopatric speciation. Allopatric speciation is a genetic divergence permitted by geographical isolation. This process is dependent on random mutation which accumulates steadily after a population has been subdivided in space. This subdivision can take place because of geographical barriers such as mountain ranges and bodies of water, because the two subpopulations are then reproductively isolated. Each accumulates different mutations and the two populations diverge. Original population is here. Initial step of speciation, barrier is formed. Then evolution of reproductive isolation. In isolation, new distant species after equilibrium of new ranges. Then parapatric, new niche entered in adjacent niche. Then sympatric, genetic polymorphism within the population. So the three general mechanisms of speciation. In allopatric speciation, reproductive isolation evolves after the population has been geographically divided. In parapatric speciation, reproductive isolation involves when a segment of the population enters a new niche. In sympatric speciation, reproductive isolation evolves within the incident group is still in the vicinity of the parent population. This divergence is strongest when the population size of one or both of the subpopulations is small, because sampling error is greatest in smaller populations. This difference will eventually lead to reproductive incompatibilities that will keep the population distinct. One well-known example of allopatric speciation involved divergence in Drosophila populations in Hawaiian archipelago. So, reproductive isolating mechanisms usually originate incidentally in the speciation process, that is, they arise incidentally during the process of evolution in isolated populations rather than being selected for. When isolated populations can come together again, incomplete isolating mechanisms may allow hybrids to form. If the hybrids are normal and viable and can freely interbreed with individuals of each parent population, then no speciation has taken place. However, if the hybrids are at a disadvantage, natural selection may favor stronger isolating mechanisms. In this case, organisms that mate with individuals from the other populations leave fewer offspring. This result is a more effective barrier to hybridization. Regions in which previously isolated populations come into contact and produce hybrids are called hybrid zones. Sympatric speciation. Sympatric speciation conversely does not require geographic isolation. Instead, it relies on the development of reproductive isolation mechanisms to allow divergence of the two subpopulations. This reproductive isolation can be a result of a number of factors, including competition for resources, disruptive selection, and sexual selection. In sympatric speciation, selection acts against individuals of an intermediate type, either through decreased viability or decreased fecundity. This creates the evolution of mating preferences and other mechanisms that result in decreased hybridization. Sympatric speciation, conversely, does not require geographic isolation. Instead, it relies on the development of reproductive isolation mechanisms to allow divergence of the two subpopulations. This reproductive isolation can be a result of a number of factors, including competition for resources, disruptive selection, and sexual selection. In sympatric speciation, selection acts against individuals of an intermediate type, either through decreased viability or fecundity. This creates the evolution of mating type, either through decreased viability or decreased fecundity, and preferences and other mechanisms that result in decreased hybridization. This sympatric speciation selection enhances traits that promote the divergence of the populations. This is fundamentally different from allopatric speciation, where mutation passively leads to differences that allow divergence of the population. Probably the best known example of sympatric speciation is the divergence of Rhagoletis pomeonella, the maggot fly. This species has recently diverged into two subspecies due to the introduction of apple trees in the northeastern United States, where hawthorn trees were native. Initially, the flies used in the hawthorn flies to reproduce on and to lay their eggs. The introduction of the apple trees provided a more nutritious food source for developing maggots, as well as the escape from parasitic wasps. Differences between the two types of fruit, such as maturation timing, allowed for the evolution of isolation mechanism and subsequent divergence to the new species. Next is about parapatric speciation. Parapatric speciation is a form of speciation in which the evolution of reproductive isolating mechanisms cross when a population enters a new niche or habitat within the range of the parent species. Generally, this form of speciation in which the evolution of reproductive isolating mechanisms occurs when a population enters a new niche or habitat within the range of a parent species. Generally, this occurs when there has been a drastic change to the environment within the original species habitat. In parapatric speciation, there is no specific extrinsic barrier to the gene flow. The population is continuous, but nonetheless, the population does not meet randomly. Individuals are more likely to meet with their geographic neighbors than individuals in a different part of population range. In this mode, divergence may happen because of reduced gene flow within the population and varying selection pressures across the population range. Evolutionary forces involved in speciation. Because genetic changes lies at the heart of speciation, the formation of new species involves the action of the same evolutionary forces known to cause genetic changes within population. The principal evolutionary forces most commonly involved in species divergence are natural selection, genetic drift, sexual selection, and mutation. All of these forces act to change allele frequencies within populations over time, and there is no question that they operate during speciation as well. First, natural selection. Darwin was the first to popularize the idea that speciation could readily occur through the prolonged action of natural selection, the process by which genetic variants that are better suited to the natural environment increase in frequency, while variants that are not well suited decrease. Then genetic drift. Genetic drift is the random change in allele frequencies caused by sampling error across generations in a finite population. Small population size can exert a strong force on the evolution of populations through genetic drift. While it occurs in all populations, drift can be major driving force for changing allele frequencies in small populations. If small populations subjected to drift, the rate of evolutionary change can be speeded up dramatically, and allele and genotype frequencies can change unpredictably from one generation to the next. The smaller the population, the more extreme these fluctuations tend to be. The actual change in allele frequency caused by genetic drift is random for any given generation. If small population subjected to drift, the rate of evolutionary change can be speeded up dramatically, and allele and genotype frequencies can change unpredictably from one generation to the next. The smaller the population, the more extreme these fluctuations tend to be. The actual change in allele frequency caused by genetic drift is random for any given generation. However, the effects of drift will accumulate over time. Much of the mathematical theory describing the process of genetic drift was developed by Sewell Wright. Like selection, drift is a process of differential reproductive success. However, the key element of selection, drift is a process of differential reproductive success. However, the key element of genetic drift is that which individuals survive and reproduce is random. unrelated to their phenotype and genotype. Then principles of genetic drift. Genetic drift, that is variation in relative genotype or allele frequencies from one generation to the next, is the result of random sampling of alleles in finite populations. If population size is small or become reduced, a small population of the total gamete pool will actually end up forming individuals in the next generation, and thus the genetic makeup of the population. Small samples taken from the gamete pool may not be representative of the range and relative frequencies of the alleles existing in the genetic pool. As a result, allele frequencies may change dramatically from one generation to the next. The magnitude of these fluctuations tend to be longer in smaller populations, and the effect of this drift are cumulative. Because it is a random process, the outcome changes in allele frequencies in any one generation is unpredictable. However, we can make certain generalities and reliably predict the cumulative effects of drift. Genetic drift has several important effects on evolution, like drift reduces genetic variation in populations, particularly reducing a population's ability to evolve in response to new selective pressure. Genetic drift acts faster and has more drastic results in small populations. This effect is particularly important in rare and endangered species. Genetic drift can contribute to speciation. For example, a small isolated population can diverge from the large population through a genetic drift. Then sexual selection. Adapting to the mutual environment is only one manner in which organisms can increase their ability to leave more offspring for future generations. Another method is to improve their ability to secure mates through this action of sexual selection. Sexual selection arises from differences in reproductive success caused by competition over mates, because variance in reproductive success can often be quite high, especially for males. So, adaptation for mating environment can sometimes be even more effective in promoting change than adapting to the natural environment, and has resulted in the rapid divergence of traits related to reproduction, such as morphology of the male genitalia, sperm composition and abundance, and male secondary sexual characteristics. The presence of the pressure for sexually reproducing organisms to have compatible reproductive systems ensure that male and female reproductive systems will co-evolve. Then maturation. Neither selection nor mutation. Neither selection nor drift can operate in the absence of genetic variability, although quite a bit of variability can be generated just through genetic recombination and the random assortment of chromosomes during reproduction. Ultimately, spontaneous mutation is the prime source of genetic variation. Mutation is considered random with respect to environmental challenges faced by organisms, although that is not to say it is without bias, because mutations are important to the context of their effects in the phenotype of organisms. Certain phenotypic classes of mutations can be more common than other classes of mutations. The founder effect. When a small number of individuals known as founder population or marginal isolates break off and form a separate population. When a small number of individuals known as founder population or marginal isolates break off and form a separate population, perhaps in a new geographical location, the ratio of alleles in the new population may be very different from the larger previous population. This is essentially a sampling difference. The larger the subpopulation, the more likely it will resemble the parent population. A classic human example is the population found in the Pitcairn Island by several of the Boutry mutineers and some Polynesians. The unique combination of causation and pollination traits, causation and pollination traits, I'm not sure about the pronunciation, sorry, that characterizes today's Pitcairn Island population resulted from the small number of founders of the population. Similarly, the Afrikaner population of Dutch settles in South Africa is descended mainly from few colonists. Today, the Afrikaner population has been unusually high frequency of gene that causes Huntington's disease, because the original Dutch colonies just happened to carry the gene with unusually high frequency. This effect is easy to recognize in genetic diseases because, of course, the frequencies of all sorts of genes are affected by founder events. That is the founder effect. Then genetic bottlenecks. Sometimes a population, or in the worst case a species, may be reduced to a very small number. The genetic diversity of the small population may be much less than the previous population, leading to a great change in allele frequency. Often endangered species are reduced to very small numbers. The Hawaiian neem of African cheetah. Both species are reduced to very small numbers, bottlenecks. The diversity of alleles present in those small populations were much less than the previous population. The loss of variation in the populations may negatively affect the ability of the species to survive future natural selection. The nature of evolution, microevolution and macroevolution. Microevolution is defined as a change in gene frequency, that is, genetic variation due to processes such as selection, mutation, genetic drift, or even migration within a population. Evolution at this scale can be observed over short periods of time. For example, between the species of the next and the frequency of the gene for pesticide resistance and population of pests increase. Such as change might come about because natural species selection favored the gene, because the population received new immigrants carrying the gene, because some non-resistant genes mutated to the resistant version, or because random genetic drift from one generation to the next. These are some possibilities. The size of the sparrow. House sparrows were introduced to North America in 1852. Since that time, the sparrows have evolved different characteristics in different localities. Sparrow populations in the north and large bodies than sparrow populations in the south. This divergence in population is probably at least partly a result of natural selection. Larger bodies birds can often survive lower temperatures than smaller bodies birds can. So colder weather in the north probably selects for larger bodies birds, hence sparrows in colder places are now generally of larger size than sparrows in warmer locales. Then macroevolution is used to refer to any evolutionary change at or above the level of species. It means at least the splitting of a species into two, that is sclerogenesis, or the change of a species over time to another anagenesis. Any changes that occur at higher levels, such as evolution of new families, phyla, or genera, are also therefore macroevolution. Microevolution and macroevolution are different things, but they involve mostly the same process. Microevolution is the change of allele frequencies, that is genetic variation due to processes such as selection, mutation, genetic drift, and even migration within a population. There is no argument that microevolution happens. Macroevolution is an evolutionary change at the species level or higher, that is the formation of new species, new genera, and so forth. Despite their differences, evolution at both of these levels arise at the same established mechanisms of evolutionary change: mutation, migration, genetic drift, and natural selection. Next, punctuated equilibria and phyletic gradualism. Punctuated equilibria evolution by Jerks states that most sexually reproducing species will show little change for most of its geological history. Most of their geological history. When phylogenetic evolution occurs, it is localized in rare events of branching speciation called cladogenesis, and occurs relatively quickly compared to the species' full and stable duration on Earth. Punctuated equilibrium is commonly contrasted against the theory of phyletic gradualism, which states that most evolution occurs uniformly and by the steady and gradual transformation of whole lineages, anagenesis. This view of evolution is generally smooth and continuous. Are evolution and survival of the fittest the same thing? Evolution and survival of the fittest are not same things. Evolution refers to the cumulative changes in a population of species through time. Survival of the fittest is a popular term that refers to the process of natural selection, a mechanism that drives evolutionary change. Natural selection works by giving individuals who are better adapted to given set of environmental conditions an advantage over those that are not well adapted. Survival of the fittest usually makes one think of the biggest, strongest, and the smallest individuals being the smartest winners, but in biological sense, evolutionary fitness refers to the ability of survive and reproduce in a particular environment. Popular interpretations of survival of the fittest typically ignore the importance of both reproduction and cooperation. To survive but not pass on one's genes to the next generation is to be biologically unfit. And many organisms are the fittest because they cooperate with other organisms rather than competing with them. How does natural selection work? In the process of natural selection, individuals in a population who are well adapted to a particular set of environmental conditions have an advantage over those who are not well adapted. This advantage comes in the form of survival and reproductive success. For example, those individuals who are better able to find and use a food source will on average live longer and produce more offspring than those who are less successful at finding food. Inheritable traits that increase individuals' fitness are then passed to their offspring, thus giving the offspring the same advantage. If fish become amphibians through the process of evolution, then why do fish still exist? That's a beautiful question. Fossil evidence clearly shows that amphibians descended from one group of ancient fishes whose thick, bony fins gradually evolved into limb like appendages. Other species gave rise to the kinds of fish that inhabit oceans, lakes, and streams around the world today. Fish, like all living creatures, continue to evolve. This evolution is not toward a life on land, but instead towards successful use of the underwater environment. There are countless ecological opportunities underwater, which is why fish still exist. When the earliest ancestors of modern amphibians left the water, they found many new opportunities on land. As amphibians and other land creatures diversified, however, fewer and fewer opportunities existed for newcomers.